Safeguard systems of DNA. Restriction enzymes, also known as restriction endonucleases, are enzymes that recognize specific DNA sequences and cleave the DNA at or near those sequences. They are an essential tool in molecular biology and genetic engineering, allowing scientists to cut DNA molecules at precise locations. Recognition sequence. Each restriction enzyme recognizes a specific short DNA sequence, typically 4 to 8 pairs long. The sequences are usually palindromic, meaning they read the same in both directions. Cutting patterns. There are two types of cuts made by restriction enzymes: blunt ends and sticky ends. Some enzymes cut the DNA strands at recognition sequence in a way that produces blunt ends where the DNA is cut straight across both strands. Other enzymes create staggered cuts resulting in sticky ends. Sticky ends have overhanging single-stranded DNA that can form hydrogen bonds with complementary sequences, facilitating the joining of DNA fragments. So, there are two types of cuts made by restriction enzymes: blunt ends and sticky ends. Some enzymes cut the DNA strands at the recognition sequence in a way that produces blunt ends where the DNA is cut straight across both strands. Other enzymes create staggered ends resulting in sticky ends. Sticky ends have overhanging single-stranded DNA that can form hydrogen bonds with complementary sequence, facilitating the joining of DNA fragments. Then bacterial origin. Restriction enzymes were first discovered in bacteria. They act as a defense mechanism against invading viruses or bacteriophages. Bacteria have methyltransferase enzyme that modify their own DNA at the recognition sequence to protect it from being cut by the corresponding restriction enzyme. Then types and nomenclature. Restriction enzymes are categorized into different types based on their properties. The most common type is type 2, which is widely used in research and biotechnology. The names of restriction enzyme usually indicate the bacterial species from which they were isolated. For example, EcoRI is derived from Escherichia coli strain RY13, while HIND2 is derived from Haemophilus influenzae strain RD. Applications. Restriction enzymes are crucial tools in molecular biology and genetic engineering. They allow scientists to create recombinant DNA molecules by cutting DNA at specific sites and joining them with other DNA fragments. This technique is used in cloning genes, constructing plasmids, generating genetically modified organisms or GMOs, DNA fingerprinting, and many other applications. Then isoschisomers and neoschisomers. Some restriction enzymes recognize the same DNA sequence and produce the same cutting pattern. These enzymes are called isoschisomers. Neoschisomers, on the other hand, recognize the same DNA but produce different cutting patterns. Isoschisomers and neoschisomers. The restriction enzyme, restriction endonuclease, REAs, ENAs, or restrictase is an enzyme that cleaves DNA into fragments at or near specific recognition sites within molecules known as restriction sites. The term restriction enzyme originated from the studies of phage lambda, a virus that infects bacteria, and the phenomenon of host-controlled restriction and the modification of such bacterial phage or bacteriophage. Restriction endonuclease recognizes a specific rather short nucleotide sequence on a double-stranded DNA molecule called a restriction site and cleave the DNA at this restriction site or elsewhere depending on the type of enzyme. Stuart Lin and Werner Arber discovered restriction endonuclease in E. coli in the late 1960s. These enzymes get their name in the fact that they prevent invasion from foreign DNA such as the viral DNA. They cut it up. Thus, they restrict the host range of the virus. Many restriction enzymes make staggered cuts into two DNA strands, leaving single-stranded overhangs or sticky ends that can base pair together briefly. EcoR1 is the first restriction enzyme from an Escherichia coli R strain. So, restriction enzymes have to do two things: recognize a specific sequence, cut or restrict at or near the sequence. Restriction enzymes differ not only in specificity and length of their recognition sequence, but also in the nature of DNA ends they generate. Thus, some enzymes such as HPAR generate flush or blend ends, others such as EcoR1, HIND2, and PST1 generate staggered ends. For example, EcoR1 cleaves covalent by phosphodiester bonds between G and A at staggered position on each strand. The hydrogen bonds between the two base pairs between these cut sites are easily broken to generate 50 protruding ends of 4 nucleotides in length. A restriction enzyme is a protein isolated from bacteria that cleaves DNA sequence specific sites, producing DNA fragments with a non-sequence at each end. Then types of restriction enzymes. There are 3 major classes of restriction endonucleases. Their grouping is based on the type of sequences recognized, the nature of the cut made in the DNA, and the enzyme structure. Type 2 and 3 restriction endonucleases are not used for gene cloning because they cleave DNA at sites other than the restriction sites and thus cause random cleavage patterns. In contrast, type 2 endonucleases are widely used for mapping and reconstructing DNA in vitro because they recognize specific sites and cleave just at these sites. In addition, type 2 endonuclease and methylase activity are usually separate single subunit enzymes: type 1 restriction enzyme, type 2 restriction enzyme, and type 3 restriction enzyme. First, type 1 restriction enzyme. The type 1 restriction enzymes are also called restriction endonuclease. They are made up of 2 long strands of DNA joined together. These restriction enzymes recognize certain sequences of DNA and cleave them at a site. The type 1 restriction enzymes was the first restriction enzyme to be identified. These enzymes are characterized by their DNA cleavage sites. Type 1 enzyme cut DNA far away from the recognized sequence in the DNA molecule. They do not cause effective fragmentation of the DNA and hence are of not much importance. The type 1 restriction enzyme have 3 subunits that perform restriction, digestion, recognition, and also modification of the DNA with the help of its cofactors like magnesium ions and ATP, adenosine triphosphate, that fulfill the catalyzing activity of the enzyme. With the type 1 enzyme, the cleavage site can be up to 1000 base pair away from the recognition site. Then type 2 restriction enzyme. Type 2 restriction enzymes are made up of 4 strands of DNA, 2 on each side of the DNA. They usually recognize 2 bases on one strand of DNA and cleave the DNA. The type 2 restriction enzymes are vastly different from type 1. For type 2 restriction enzymes, recognition of the sequence and restriction digestion DNA cleavage occurs at the same place. The sites are not different from each other. Moreover, for the cofactor, the type 2 restriction enzyme usually only use magnesium ions for completing the restriction process in DNA molecule. The type 2 type is the most common restriction enzyme available and is used in the most for carrying out restriction. Another major characteristic of the type 2 enzyme is that these enzymes either cut through the middle of the DNA strand, causing blunt ends at both sides, or create cleaves at staggered positions, leaving sticky ends. The type 2 restriction enzymes also have more than one subunit, and the subunits perform different functions. These are a family of unrelated proteins. They are named after the bacterial species which they are isolated. For example, EcoR1 is isolated from bacterial species E. coli. For example, the type 2 enzyme is called EcoR1, isolated from E. coli, cuts DNA only at the hexanucleotide given. So, digestion of DNA with a type 2 enzyme therefore gives a reproducible set of fragments whose sequences are predictable if the sequence of the target DNA molecule is known. The restriction enzyme generates 2 different types of cuts. Blend ends are produced when they cut the DNA at the center of recognition sequence, and sticky ends produce an overhang. Type 2 enzymes are distinguished because the recognition site and cleavage site are the same. These sites range in length from 4 to 8 base pairs. The sites are typically inversely palindromic, that is, reading the same forward and backward on complementary strands. Restriction enzymes can cut the DNA in 2 different ways as demonstrated later. The first and the most common is staggered cut, which leaves single-stranded overhangs or sticky ends. The overhang can be 3' or a 5' overhang. The second way is the blunt double-stranded cut, which does not leave an overhang. An additional level of specificity determines whether an enzyme will cut DNA containing a methylated base. The degree of specificity in the site also varies. Most enzymes are very specific, whereas some will allow multiple bases at one or two positions within the site. Type 2 restriction endonuclease recognize and cleave DNA within particular sequences of 4 to 8 nucleotides which have a twofold axis of rotational symmetry. Such sequences are often referred to as palindromes because of their similarity to words that read the same backward and forward. For example, the restriction and modification enzymes EcoR1 and MEcoR1 recognize the sequence next axis of symmetry. Type 3 restriction enzymes. These are multifunctional proteins with two subunits, res and mod. It is a modification methyl transferase. The DNA sequence specific for the system is recognized by the MOD subunit. Type 3 restriction enzymes are DNA cutting enzymes that recognize patterns of DNA that are not necessarily based on DNA sequence. Type 3 restriction enzymes are multifunctional proteins. This type of restriction enzyme cut the DNA away from the recognition sequence. They have two subunits that carry the function of DNA methylation or modification and restriction digestion. These enzymes use the adomet cofactor generally for carrying out the restriction process. Type 3 factors enzymes cleave at sites a short distance from the recognition site, require ATP but do not hydrolyze it. Type 3 enzymes have closer cleavage sites, usually 20 to 30 base pair away. Then, major kinds of damage to DNA and causes. Damage to DNA consists of any change introducing a deviation from the usual double helical structure. There are three major classes of DNA damage: single base changes, structural distortion, and DNA backbone damage. Damage to DNA consists of any change introducing a deviation from the usual double helical structure. There are three major classes of DNA damage: single base change, structural distortion, and DNA backbone damage. DNA can be damaged in many different ways, and this damage, if left unrepaired, can lead to mutations. Changes in the base sequence of a DNA. This distinction is worth emphasizing at the onset. DNA damage is not the same as mutation, although it can lead to mutation. DNA damage is simply a chemical alteration to DNA. A mutation is a change in a base pair. For example, the change from a GC pair to an ethyl GC pair is DNA damage. The change from a GC pair to any other natural base pair, ET or TA or GC, is a mutation. So, if a particular kind of DNA damage is likely to lead to a mutation, we call it genotoxic. Single base changes. A single base change or conversion affects the DNA sequence but has only a minor effect on overall structure. For example, replacement of the amino group of cytosine with oxygen converts cytosine to uracil, a base that should only be present in the RNA chain. This type of conversion process is called deamination. Deamination is the most frequent and important kind of hydrolytic damage and can occur spontaneously from the action of water or may be induced by a chemical mutagen. When the UG base pair replaces the CG base pair, this causes only a minor structural distortion in the DNA double helix. This type of damage is not likely to completely block the process of replication or transcription, but may lead to the production of a mutant RNA or protein product. Vertebrate DNA frequently contains 5-methyl cytosine in place of cytosine. Methylated cytosines are hotspots of spontaneous mutations in vertebrate DNA because deamination of 5-methyl cytosine generates thymine. This results in the change of a GC base pair into an AT when damaged DNA is replicated. DNA also subject to damage by alkylation, oxidation, and radiation. Alkylating agents such as nitrosamines lead to the formation of O6-methylguanine. This modified base often mispairs with thymine, resulting in the change of a GC base pair into an AT base pair when the damaged DNA is replicated. Potent oxidizing agents are generated by ionizing radiation and by chemical agents that generate free radicals. These reactive oxygen species, for example O2H2O2OH, can generate 8-oxoguanine, a damaged guanine base containing an extra oxygen atom. OXO-G is highly mutagenic because it can form a Hogsteen base pair with adenine. This gives rise to a GCTA conversion, which is one of the most common mutations found in human cancers. Then structural distortion. Ultraviolet light has a detrimental effect on cells due to selective absorption of the UV rays. Radiation with a wavelength of about 260 nm is strongly absorbed by the bases. The most frequent UV-induced lesions of DNA are the induction of pyrimidine dimers between two neighboring thymine bases by UV radiation. These are also termed cyclobutane pyrimidine dimers because a cyclobutane ring is generated by links between carbon atom 5 and 6 of adjacent thymines. Because covalent bonds form between thymines on the same strand, this disrupts the complementary base pairs that form the duplex helix. Thymine dimers thus distort the structure of the duplex DNA. Structural distortion may impede transcription and replication by blocking the movement of polymerases. Consequently, the induction of pyrimidine dimers is a more severe defect than a single base change. UV radiation can also induce dimers between cytosine and thymine, called pyridine photoproducts. Other bulky adducts can be induced by chemical mutagenesis by exposure to large polycystic hydrocarbons or alkylating agents. DNA damage is also caused by intercalating agents and base analogues. Intercalating agents such as ethidium bromide contain several polycystic rings. These flat rings insert between the DNA base, binding and stacking with the DNA bases, just as the bases bind or stack with each other in the double helix. Due to the resulting distortion of the double helix, intercalating agents can cause insertion or deletion of one or more base pairs during DNA replication. Base analogues are compounds such as 5-bromouracil, an analog of thymine that substitute for normal bases. They are similar enough to the normal bases to be taken up by cells, converted into deoxynucleotides, and incorporated into DNA during replication. However, because of their structural differences, analog base pair inappropriately. For example, 5-bromouracil can mispair with guanine. Next, DNA backbone damage. Backbone damage includes the formation of abasic sites, loss of the nitrogenous base from a nucleotide, and double-strand DNA breaks. Abasic sites are generated spontaneously by the formation of unstable base adducts. For example, in purine nucleotides, the sugar-purine bonds are relatively labile. Hydrolysis of N-guanosyl linkage in the purine base by the action of water leaves a hydroxyl OH in its place in the depurinated DNA. Double-strand breaks can be induced by ionizing radiation, X-rays, radioactive materials, and a wide range of chemical compounds. Ionizing radiation can attack or ionize the deoxyribose sugar in the DNA backbone directly or indirectly by generating reactive oxygen species. Double-strand breaks are the most severe type of DNA damage since they disrupt both DNA strands. Damage caused by alkylation of bases. Alkylation is the reaction of electrophilic chemical compounds or alkylating agents with nucleophilic centers in organic macromolecules. Alkylating agents include a large variety of chemicals, some of which are potent mutagens or carcinogens. Alkylating agents include the large number of molecules which can effectively react with DNA, causing structural modification of the base and phosphate group in DNA. The most common agents are alkyl sulfates, alkyl sulfonates, alkyl halides, dialkyl nitrosamine, alkyl nitrosources, acyl nitrosamines, mustards, diazo compounds, lactones, epoxides, etc. These agents can be divided into two major groups, monofunctional and bifunctional. The monofunctional agents have only one reactive group which is involved in covalent interaction with a single center on DNA. The bifunctional agents have two reactive groups and have an ability to react with the two centers on DNA. If the two centers are on the opposite strands of the DNA, the reaction of a bifunctional agent such as sulfur or nitrogen mustards can produce an intrastrand crosslink. Any of the alkylating agents have a mutagenic effect and known as suspected carcinogens. The reaction specificity of these compounds is differed from different bases. Diazoalkyls react readily with guanosine and thymidine, but only under extreme conditions with aldosine and cytosine. In contrast, alkyl sulfates, alkyl sulfonates, mustards, epoxides, and nitroso compounds, alkylate bases in the following order: GACT. The rate of ethylation is much slower than that of methylation or for all classes of alkylating agents except alkyl iodides, which readily alkylate Ga and C. Many environmental carcinogens or cancer-causing agents are electrophiles that act by attacking DNA and alkylating it. As we have just seen, this can lead to mutations. If the mutations occur in genes that control the other or otherwise influence cell division, they can cause a cell to lose control over its replication and therefore change into a cancer cell. Damage caused by ultraviolet radiation. Ultraviolet radiation cross-links adjacent pyrimidines on the same DNA strand, forming two major lesions. 80 to 90% of these are pyrimidine dimers, which are also called cyclobutane pyrimidine dimers because of the four-membered cyclobutane ring that forms between the two bases. 10 to 20% of the lesions are 6,4 photoproducts, in which the 6 carbon of the pyrimidine is linked to the 4 carbon of the adjacent pyrimidine. Each of these products block DNA replication because they are non-informative or non-coding. The replication machinery cannot tell which bases to insert opposite the lesion. As we will see, replication sometimes proceeds anyway. The bases are inserted without benefit of the base pairing that normally provides accuracy. If these are the wrong bases, the mutation results. Then damage caused by gamma and X-rays. The much more energetic gamma rays and X-rays, like UV rays, can interact directly with the DNA molecule. However, they cause most of their damage by ionizing the molecule, especially water surrounding the DNA. These ions, free radicals, chemical substances with an unpaired electron. These free radicals, especially those containing oxygen, are extremely reactive and they immediately attack neighboring molecule. When such a free radical attacks a DNA molecule, it can change a base or it can cause a single or double stranded break. DNA bases are subject to at least 20 kinds of oxidative damage, and this can be caused by reactive oxygen species derived from ionizing radiation or simply from normal oxidative metabolism. The best studied oxidatively damaged DNA bases O-oxoguanine, also known as 8-hydroxyguanine. DNA polymerase in bacteria and eukaryotes misread OXO-G as thymine and insert adenine instead of cytosine, resulting in an OXO-G pair. Both bases in this pair are genotoxic because they both will probably lead to mutations if they are not removed before the DNA replicates again. Single-stranded breaks are ordinary but serious because they are easily repaired, just like rejoining the ends of several severed strands. But double-stranded breaks are very difficult to repair properly, so they frequently cause elastic mutation. Because ionizing radiation can break chromosomes, it is referred not only as mutagen or mutagen-causing substance, but also as a clastogen, which means breaker. Repair mechanisms of DNA. DNA repair mechanisms act to prevent mutation and necrobiosis, thus maintaining genome stability, cellular integrity, and preventing the onset of cancer, neurodegeneration, and premature aging. Direct reversal. Photoreactivation repair refers to the DNA repair system where the X exposure repairs the UV damaged DNA under the visible light. It is also called the direct reversal DNA repair mechanism, in which the photolysis enzyme under action of visible light reverses the effect of UV radiation in the cells. Photoreactivation repair. DNA with lesion, normal DNA without dimer. Pyrimidine dimer, photoreactivation by visible light exposure to UV radiation. UV exposure, dimer formation. Mechanism of photoreactivation. The photoreactivation process repairs the UV damaged DNA. A DNA helix becomes distorted and forms a pyridine dimer, usually thymine-thymine dimer, under UV light exposure. A dimer forms when the adjacent thymine bases attach covalently to their respective C5 and C6 atoms. The formation of TT dimer causes helix distortion, which eventually causes replication errors in the DNA and finally leads to mutation. The covalent bond formed between the two adjacent thymine bases must be broken to recover the helix distortion. An enzyme proteolysis breaks off the covalent bond between the dimer. Then formation of pyridine dimer. The UV light induces a covalent bond between the pyrimidine bases, either thymine or cytosine. The two adjacent pyrimidine bases bind through a carbon-carbon double bond. A carbon-carbon double bond formation between the pyrimidine base form a dimeric structure commonly referred to as cyclobutane pyrimidine dimers. Cyclobutane pyrimidine dimers is a common product forming under the UV exposure. In addition to this, 6 to 4 photoproducts also produced as a result of UV damage. There are two bases attached with a single carbon bond to the C6 of one ring and C4 of another ring. Thymine-thymine dimer usually forms after this UV exposure and it appears as a bubble in the DNA strand. Treatment with photolysis under visible light. Under the visible light of a long wavelength, a proteolyase enzyme becomes activated. A proteolyase enzyme scans the DNA strand and recognizes the pyrimidine dimers. After recognition, the dimer it forms in the DNA, it directly binds to that segment. Breaking of TT dimer. When a proteolyase enzyme binds to the TT dimer, it starts splitting the carbon-carbon double covalent bond between the cyclobutane ring. Thus, a proteolyase enzyme converts the dimeric form of pyrimidine base into monomeric forms. Photo reactivation repair. TT dimer covalent bond DNA strand. Act directly on damaged nucleotides, converting each back to its original structure. Only some kinds of damaged nucleotides can be repaired directly. Nicks can be prepared by DNA ligase if all that has happened is that a phosphodiester bond has been broken without damage to the 5' phosphate and 3' hydroxyl groups of nucleotides either side of the nick. It is often the case with nicks resulting in the effects of ionizing radiation, alkylating damage. Some forms of alkylation damage can be directly reversed by enzymes. An enzyme MGMT or AGT repairs two varieties of DNA adducts, methylguanine and methylamine. Mismatch repair. DNA mismatch repair is a system of recognizing and repairing errors, insertion, deletion, and misincorporation of bases that can arise during DNA replication and recombination, as well as repairing some forms of DNA damage. Mismatch repair is strand specific. During DNA synthesis, the newly synthesized daughter strand will commonly include errors. In order to begin repair, the mismatch repair machinery distinguishes the newly synthesized strand from the template. Examples of mismatched bases include a GT or AC pairing. Mismatches are commonly used to tautomerization of bases during DNA replication. The DNA mismatch generates due to the incorporation of an incorrect base during the DNA replication process. The mismatch repair system uses the methylation on the parental strand as a signal to leave that strand alone and to correct the nearby mismatch in the unmethylated progeny strand. The nicked DNA segment is then excised by the EXO1 exonuclease. Mismatch repair depends on the number of activities in human cells, including MUTs alpha, continuation will be dealt with the help of next video.